안녕하세요. 저는 MIT에서 생명공학 PhD 캔디데이트로서 공부하고 있는 김영훈이라 합니다. 브릭에서 이렇게 웨비나 초청해 주신 걸 너무 영광스럽고 저의 연구 결과 발표할 기회 주셔서 너무 감사합니다. 저는 지금 미국 케임브리지 매사추세츠에 있는 MIT와 와이헤드 인스티튜트에서 진카 웨인 교수 연구실에서 식물 관련된 연구를 하고 있습니다. 오늘 발표한 토픽은 다중오믹스 인포메이션을 이용한 식물 할로게나제 발견과 엔지니어링 관한 내용입니다. 제가 한국말이 좀 부족해서 영어로 프레젠테이션 시작하겠습니다. Thank you all for coming to my webinar. I'll be sharing my research on multi-omics guided discovery of plant halogenase in Menisperm ACA plants. To give a general background of my research, plants have evolved for millions of years to produce specialized metabolites and developed highly specialized enzymatic pathways to support their biosynthesis. Currently, approximately 1/3 of all existing pharmaceuticals are of plant origin. And to showcase some of these examples, here are vinblastine and paclitaxel, which are anti-cancer drugs found in Madagascar periwinkle and Pacific yew tree. Other examples include colchicine, which is an anti-inflammatory drug for treatment of gout that is found in flame lily plants, and also pain relief opioids like morphine and opium poppy. These drugs are extremely valuable, and most of these natural compounds are directly isolated from native plants, which the process is actually very laborious in practice and is often limited by slow and variable plant growth. An alternative strategy is to synthesize these compounds using organic chemical synthesis approach. But this approach is also extremely costly and produces toxic waste, which is harmful for the environment. Also, many of these compounds involve large numbers of steps in chemical synthesis, which is not efficient compared to natural biosynthesis. With the recent characterization of these biosynthetic pathways of these plant natural compounds, metabolic engineering in heterologous hosts like baker's yeast has greatly presented a cost-efficient alternative to producing these compounds, and a greener approach, especially for tackling climate change. But metabolic engineering heavily relies on the elucidation of the biosynthetic pathways of these plant natural products, which many of them still remain under-characterized. Therefore, characterizing these biosynthetic pathways will greatly advance our biochemical understanding of how plants synthesize these compounds with great structural complexity. And it will also enable tools in synthetic biology to produce them in commercial-friendly heterologous hosts. Here in our lab, I'm specifically interested in the medicinal alkaloids that are found in the plant vines of Menisperm ACA family. The Menisperm ACA family belongs to the Ranunculales order and is a sister to other families that have been well studied, like the Papaver ACA family that produces morphine, a pain relief drug that was mentioned in the previous slide. While its neighboring families have been widely investigated for their natural product biosynthesis, the alkaloids found in the Menisperm ACA family remain underexplored. So I'll quickly share the broader background of the different types of alkaloids found in this family and zoom into a specific one that I'll be focusing on for this talk. The plants in the Menisperm ACA family are mostly climbing vines, which are portrayed in the middle here, and have been used as herbal remedies for centuries to treat various medical conditions ranging from dysentery to dermatitis. And there are three main different interesting categories of medicinal alkaloids that are found in this family. The Acutamine type, the Hasubanan type, which both are derived from the benzoyl isoquinoline alkaloids, abbreviated as BIA in this presentation. And the third category being the bis-benzoyl isoquinones, which harbor the two BIA backbones connected by a phenylcoupling reaction. These compounds have shown to exhibit potent medicinal properties like anti-cancer, narcotic antagonistic, and antiviral activities. While many of these compounds are interesting in its structure and also with their unique bioactivities, today I'll be focusing on Acutamine, which are mainly produced in the menispermum and cinnamonium vines. Acutamine is a rare chlorinated plant natural product and harbors a tetracyclic alkaloid structure. It has been shown to exhibit selective cytotoxic activity towards human cultured T cells, along with interesting memory-enhancing activities when tested in Wistar rat models. I chose to specifically focus on acutamine because its biosynthetic pathway is speculated to involve very rare enzymatic transformations, including this regio and stereo selective halogenation at this otherwise unactivated carbon center, which is a valuable biochemical reaction seen in many drug compounds that harbor these halogen molecules. Halogen is a common functional group found in many different therapeutical drugs and is often essential for the observed bioactivity. These are some of the examples of halogenated drugs, like chloramphenicol, which is the first antibiotic drug that was mass-produced in large scale, and shringomycin E, which is a lipocyclic peptide used as an antifungal drug or a detergent to dissolve plant membranes. And another popular antibiotic used against C. diff infections called vancomycin, which is a pretty big structure. And these halogenated compounds are naturally found in bacteria, and many others are also commonly found in marine animals and macroalgae. Despite more than 5,000 of these characterized halogenated natural products to date, not many have been actually identified in land plants, acutamine being one of the handful of the plant halogenated compounds found in nature. If you take a closer look at this molecule, you can recognize that it contains an sp3 carbon halogen bond, which is extremely difficult to install in a site-specific manner, especially at the late stages of a chemical synthesis. This biochemical transformation involving regio and stereoselective halogenation is of great interest in the medicinal chemistry, as the bioactivities and the pharmacokinetics of drugs can often be fine-tuned through halogenation. Here, I speculated that a specific class of halogenase enzyme called the iron 2,2-oxoglutarate dependent halogenase, abbreviated as 2ODH, was catalyzing the site-selective CH halogenation in acutamine biosynthesis. 2ODHs belong to a bigger family of enzymes called the iron 2,2-oxoglutarate dependent dioxygenases. It's a mouthful, but it is the second largest family of oxygenases after the cytochrome P450s. And a canonical 2ODDs activate molecular oxygen using a ferrous cofactor and a 2-oxoglutarate cofactor to oxidize a aliphatic CH bond into a hydroxyl outcome, for example. These 2ODDs can catalyze a diverse range of reactivities, with hydroxylation being the most common, but many others, including radical group transfer, stereoinversion, endoperoxidase formation, and a series of different desaturation reactions, and many other reactions that haven't been mentioned in this presentation. The mechanism and the structure of these enzymes are very interesting, as the canonical hydroxylases have a highly conserved iron-binding facial triad with histidine and aspartic acid, along with a distal histidine and a 2-oxoglutarate in the active site that coordinate the iron. In the previously characterized bacterial 2ODH halogenases, we observed a mechanistic active site substitution where this aspartic acid residue is replaced by a small amino acid like glycine or alanine, which allows this halide anion to occupy an iron coordination site and in turn elicits halogenation chemistry. This mechanism is truly fascinating, since depending on what is coordinating this ferrule intermediate, it is able to catalyze the CH abstraction to generate a radical in this carbon, and then allow for installation of a hydroxyl group or an anion group onto an otherwise unactivated carbon center. With this mechanistic insight and the fact that other halogenase enzymes like haloperoxidases and SAM-dependent halogenases do not exactly have the same kind of capability to install a halogen onto an unactivated carbon center. I specifically searched for 2ODH halogenase genes in my study. And I searched for these candidates using a multi-omics guided approach. Here, I'll give you an overview of the method that was applied in identifying the plant halogenase involved in the terminal chlorination of Acutamine biosynthesis. With the recent advances in sequencing and multi-omic analysis tools, I used a combination of metabolomics to profile the tissue-specific metabolites in the plant of interest, and I would combine it with transcriptomics by tissue-specific RNA sequencing and analyzing the highly and differentially expressed genes in the tissues of the plants that are relatively accumulating the compound of interest. This process generates a list of candidate genes, which can be narrowed down by building a maximum likelihood phylogenetic tree to infer any evolutionary information about the candidate. If the candidate genes fall under the same clade that only consists of producing plants, then they are more likely to be specialized enzymes participating in the biosynthetic process that is unique to those organisms. And from this list of select halogenase candidates, I recombinantly expressed the halogenase enzyme in E. coli and purified to examine its in vitro biochemical functions using enzyme assays and liquid chromatography and high resolution accurate mass spectrometer, LCMS, to analyze really the outcome and the functions of these halogenase candidates. Now that I've described the general workflow of this study, I'll discuss about the specific example and the recently published study on the identification of dichloroacutamine halogenase. First, we obtained 3 different species of the menisperm ACA plants. 2 known acutamine producers, as previously mentioned, the M. canadense and S. acutum species, and 1 non-producer, Stephania japonica, which belong to the same family but are devoid of the acutamine type alkaloids. And we performed untargeted metabolomics using high-resolution LCMS to perform tissue-specific metabolic profiling and to detect the production of acutamine type alkaloids. Here on the top right, you'll see 6 different acutamine type alkaloids that were detected in the LCMS extracted ion chromatograms. On the bottom here are the LCMS extracted ion chromatograms that correspond to the mass to charge ratio of these alkaloids. Each of these chromatograms correspond to a specific tissue type in the species that were assayed. Here on the x-axis is the retention time from the LC separating the different compounds, and the y-axis is the ion count and the intensity values of the compounds detected by the mass spectrometer. As expected, here in blue, we observed no acutamine type alkaloids in the non-producer Stephania japonica roots, while the other 6 alkaloids were detected across the root and the leaf tissues of the producing plants. Analyzing this rich metabolic data set, we were mainly interested in whether acutamine is accumulated in a tissue-specific and a species-specific manner. And we found out that while acutamine was detected across multiple tissue types in both M. canadense and S. acutum, its accumulation is relatively more enriched in the roots of M. canadense compared to other tissues that were profiled in this experiment. With this knowledge, we performed tissue-specific RNA-seq experiments, followed by de novo transcriptome assembly for each of these menisperm ACA family of plants to enable halogenase gene discovery. To analyze our assembled transcriptomes of these plants, we first looked at the biosynthetic proposal of acutamine and determined whether if there are signatures of already known genes that can be used to assess the data set for gene discovery. Here is a biosynthetic proposal starting from L-tyrosine, an amino acid from a primary metabolism, all the way to acutamine, our compound of interest. These genes that are involved in the biosynthesis of a common BIA precursor, S-reticulin, have been found in the resulting de novo transcriptomes of those acutamine-producing plants. In fact, here is a transcript differential expression in M. canadense root versus leaf tissues, with the x-axis and the y-axis denoting the transcript abundance quantified by transcript per million values in log scale. Each dot here represent a transcript sequence with some of these big colored dots representing genes that are proposed to participate in the acutamine biosynthesis. And interestingly enough, in addition to the BIA biosynthetic genes that were highlighted here, according to the color, we also found a transcript colored here in red that exhibits a 459-fold higher transcript expression level in the roots of M. canadense compared to that of in leaves, which was consistent with the previous metabolic analysis that the acutamine accumulation was the most enriched in the root tissues. Using these two omics data sets, we discovered that this highly and differentially expressed gene and named it as dichloroacutamine halogenase, and I'll be referring to this gene as DAH in this presentation. We looked closer at the sequence of this DAH gene and found orthologous sequences in both S. acutum and M. canadense transcriptomes, which are the both two acutamine producing plants, with 99.1% sequence identity at the protein level. Then we aligned these sequences with other plant 2ODD hydroxylase sequences and discovered the iron binding facial triad motif that I mentioned before. We found out that the DAH gene harbored this active site aspartic acid residue mutation to glycine, which was consistent with the previously identified halogenase, where the mechanistic substitution allowed for a halide ligand coordination to the iron and resulted in halogenation chemistry. This gave promising insights for us to move forward with the characterization experiments. We further analyzed the DAH gene and assessed its evolutionary origin by constructing a maximum likelihood phylogenetic tree using select plant 2ODD sequences, along with already characterized bacterial 2ODH halogenase sequences. Here you can see at the top that the 5 selected bacterial halogenases form a distinct clade, whereas the DAH gene form a cluster together with other plant dioxygenase genes. This analysis suggested that DAH evolved independently from those of the 2ODH halogenases previously identified from bacteria. And to the best of our knowledge, this discovery of DAH represents the first 2ODH of plant origin, which is a remarkable finding as there is only a handful of 2ODHs that have been characterized, and all of them had been bacterial halogenases. This tree also shows that DAH may have evolved from ancestral plant 2ODDs, more specifically the FLS-like, the flavanol synthase-like progenitors, through a gene duplication event in the radiation of ranunculales that gave rise to the specialized acutamine biosynthesis in Menispermaceae plants. We then biochemically characterized the function of the DAH gene, with our hypothesis being that it is involved in the coordination of dichloroacutamine to produce acutamine. We carried out the enzymatic assay of the wild-type DAH under previously defined 2ODD activity assay conditions and analyzed using high-resolution LCMS. Here you see on the x-axis is the retention time in minutes, showing the separation of the compounds detected in the LCMS, and y-axis showing the relative intensity of the extracted ions. On the bottom here is the -2 oxoglutarate control, which shows a clear peak corresponding to the substrate dichloroacutamine. And on the top here in blue is the +2OG control, which shows a clear conversion of the substrate into acutamine at a distinct retention time. The MS2 fragmentation patterns, which are not shown here, also were used to validate the identity of these peaks. And from this assay, we confirmed that the terminal chlorination function of wild-type DAH showed the conversion of dichloroacutamine to acutamine in the biosynthesis in menispermaceae plants. After confirming the biochemical function of DAH, we wanted to assess the role of the potential mechanistic active site mutation found in DAH relative to canonical 2,0-DD hydroxylases. So we explored a glycine to aspartic acid mutation, which is the opposite, to replace the chloride anion with the acidic residue in the active site to coordinate the iron, and tested its activity against its native substrate, dichloroacutamine. We suspected that if DAH adopts the same mechanistic substitution as in bacterial 2,0-DHs, this mutant would restore the canonical hydroxylation activity to form 11-hydroxy-dichloroacutamine in a site-specific manner. In fact, the mutant enzyme assay did exhibit this hydroxylation outcome. Here in black is the negative control having no 2,0-G, with the mutant showing that the large substrate peak here. And here in blue, you see the wild type assay showing the full conversion of the substrate into acutamine. And here in red is the mutant assay that was entirely devoid of the halogenase activity and instead exhibits the exclusive hydroxylation to give rise to a new peak that corresponded to the MZ value of the hydroxylation product highlighted here. This result confirmed our validation that DAH is indeed similar in its mechanistic properties, and since reverting the iron coordinating facial triad with the acidic residue restored its canonical hydroxylation activity instead of the halogenation outcome. Next, I want to highlight the engineering potential of DAH, as previously characterized bacterial 2,0-DHs have also been shown to install alternative anions other than its native anion. As mentioned in the mechanism of these 2,0-DHs, the anion that coordinates the iron here reacts with the activated carbon radical to functionalize the sp3-CH bonds. And therefore, I set out to test a panel of alternative anions and tested the enzyme assays replacing the sodium chloride salt with their respective salts. And out of all the anions tested, like bromine, nitrite, cyanate, and azide, only azide resulted in a derivatized product, which was identified as 11-azido-dichloroacutamine. And here you can clearly see that if you replace the anion with the sodium azide in the enzyme assay condition, you'll see a rise of a new peak corresponding to the azido product. And you'll also see the presence of acutamine due to the trace chlorine anion that gets carried over in the enzyme buffer, and also a hydroxylated product due to an elevated hydroxylation outcome when the enzyme access to its native chlorine substrate is limited. This finding is fascinating, as there is no naturally occurring enzymes that can catalyze azidation. And this new to nature azidation capability of DAH opens new avenues for click chemistry based derivatizations. Also, this shows the potential for DAH as a starting point for expanding anion selectivity to catalyze other CH functionalization outcomes. In summary, here I presented a powerful example of the combined metabolomics and transcriptomics approach for biosynthetic gene discovery with the identification of DAH in acutamine biosynthesis. And evolutionarily characterized DAH as the first 2,0-DH of plant origin, which falls under a handful of regio and stereospecific halogenases that can target unactivated sp3 carbon hydrogen bonds. And finally, I showcased the biochemical characterization of DAH as the enzyme responsible for the terminal chlorination of acutamine biosynthesis, and also explored its origin from plant 2,0-DD hydroxylases, likely the flavonol-like synthase proteins that are involved in the flavonoid biosynthesis. And I also highlighted here as DAH as a promising scaffold for engineering novel biocatalysts that can catalyze CH functionalizations like azidation on potent plant natural products to derive new-to-nature therapeutics. There's a lot of open directions that this work provides, and I'll discuss 3 interesting ongoing studies. The first direction is to engineer DAH to expand its substrate promiscuity and to evolve mutants that can halogenate BIA compounds that are precursors to many already known potent drugs like morphine and codeine. Here we can use directed evolution to design and select DAH mutants that can achieve such desired halogenation activity on these BIA structural analogs. And second, going off of the CH azidation of plant natural products, we are developing click chemistry enabled tools to further derivatize these azidated target natural product scaffolds and to produce new to nature therapeutics. And finally, another future direction of this study is to elucidate the complete biosynthesis of Acutamine type alkaloids that are of medicinal importance. This will further not only our knowledge on alkaloid biochemistry, but also enable metabolic engineering to produce these potent medicinal compounds in a more climate-friendly and efficient way. So I would like to thank all the members of the Wang Lab for their support and making my PhD experience amazing so far. And I would like to specifically highlight my PI, Jinka, for his continued support, and the DAH manuscript team, Andrew, Chris, Fushuang, and Tomash for all of their help and input. And of course, Valentina, our lab manager, for her support. I would also like to thank my thesis committee, Jim, Angela, and Chris for their input on their thesis project so far. And of course, the amazing core facilities here at the Whitehead and Koch Institute, along with the MIT Biological Engineering Department. 그리고 저를 초청해 주시고 이런 자리를 마련해 주신 브릭 관계자분과 브릭 커뮤니티한테 너무 감사합니다. 지금까지 발표 들어주신 여러분께도 감사합니다.